All right, so the first one we're going to look at is sex linkage. I'm going to share a white screen. All right, so sex-linked genes. All right, we're going to talk about two types of linkage today. One of them is this one, sex-linked genes. And in this case, we're talking about trait genes, single genes, right? Genes for single traits that happen to be located, on the sex chromosomes. On the sex chromosomes. Okay, so you know what the sex chromosomes are. For a female mammal or a female human, the sex chromosomes are X and X. Females have two X sex chromosomes, and male humans have one X chromosome and one Y chromosome. So when we're talking about particular traits that are located on sex chromosomes, we're only talking about, in our case, we're talking about that the genes are on the X chromosomes only. Okay, only on the X's. This is something called X-linked. And there is such a thing as Y-linked genes. But in terms of what we need to learn for AP biology and what you learned as freshmen, all the genes we learned about are X-linked. In other words, the alleles for those genes, or the letters that represent those genes, are located. What we do is we put them here in the corners of the X chromosomes because they're located somewhere here on the chromatids of those X chromosomes, and they're not found on the Y, right? They're not there. That X means they're not there. It's not another chromosome. I hope I'm not confusing you with that. So these are not carried on the Y chromosomes. And that is why the expression in males and females can be very different whenever the genes for those traits, whenever you're talking about traits that are sex-linked. And here's the reason why. So let's do an example question really quickly. So for example, first of all, let's just talk about the disorders quickly that are specifically sex-linked. You may remember include color blindness. That's one. Hemophilia, which is a disorder where a person has trouble clotting their blood, and that can be very, very dangerous if they are injured. There's some other things too, but those are the two main ones that you're mostly going to see are color blindness and hemophilia. Okay, so if we use this notation, now let's do a quick example. Let's just do an example problem. Let's say you have a woman who is a carrier. Carrier means you are heterozygous, okay? That's what you need to know. And you know what that is. For hemophilia. And a man who has normal blood clotting, okay? Normal blood clotting. Now that is actually enough information to do the genotypes for both parents. And here is why. So if I read this question and I want to know what my parents are, then I'm then going to put into a Punnett square, right? The woman is going to be, first of all, XX, because she has 2 sex chromosomes that are XX. If she is a carrier, and if we assume that big H is normal and little h is hemophilia, the mutated gene that prevents proper blood clotting, then she's a carrier. So one of her X's is carrying a big H and the other one is carrying a little h. There in those corners of the X's, you make them superscripts. Crossed with a man who is going to have XY. Remember, the Y doesn't carry anything. And since he has normal blood clotting, the one letter he will have on his 1X that he has is a capital H. There's the genotypes of the parents, half the battle. The biggest difference in terms of, crossing other things like you did on that worksheet where everything turned green for you, right, is that you have to put the parents' sex chromosomes first and then attach the proper alleles to the X's only based on the information the problem gives you. So now if you set up the Punnett square, the trick is just to make sure that you keep all of the H's connected to their X's as you go through the Punnett square. So if you draw a Punnett square like that. And let's put mom up here with her 2 X's and dad over here with his 1X and his Y. And then you carry the letters through as you would normally, right? So you have one daughter who is homozygous normal, one daughter who is also a carrier, one son who is normal and will have normal blood, and one son who will be a hemophiliac. So this is exactly what happened in the royal family of Europe, where Queen Victoria was this mother right here. She was a carrier, and she had a lot of babies, and many of her daughters were carriers. She actually had quite a few daughters, and those carriers were then princesses who were married off all over Europe and took that hemophilia gene and ruined the male line, the male heirs, by causing many of the sons of European royal families to be born with hemophilia, which back then was really deadly and difficult to treat. They called it the bleeding disease, right? So very, very sad thing there. Okay, so now the other thing I wanted to make sure that I pointed out, I know you're posting me questions, just give me 1 second, is when you do the outcomes for this, be really careful how they ask you the question. Because what if they asked, what percentage of the boys or the sons are hemophiliacs? What would be the answer to that? What percentage of the sons? Are you saying 50%? Because you're right. It would be 50% of the boys, right? This is 100%. So 50% of the sons would be hemophiliac. So just be really careful, because when they ask those questions, sometimes the language, you have to be very careful what they're saying. Now, on the other hand, if they said, what percent of the offspring, right? What percent of the offspring are hemophiliacs? Then, right, then you would say 25%, because if this is all the offspring, then that represents 25%. So just be careful of that, okay? Because sometimes they ask, and not just with Sexlinked, but they'll do that a lot of times. Okay, so that is what I wanted to tell you about sex linkage.